Hello everyone. Today we are here to show you something remarkable. For decades, marine researchers have gone into deep oceans collecting precious samples. But here's the truth. It takes around 6 to 8 hours to process one single sample. 85% of the deep sea organisms aren't in any database. Researchers return with thousands of life forms that science can't identify. We refuse to accept this. Today, Team GeneX is here to show you what we have built and where we are heading to revolutionize deep sea discovery.
Now, a model has been trained on 12,000 plus sequences, going through multiple filters, transformations, and augmentations. Once the data is processed, the real story begins. Now, data only matters if it creates patterns, and these graphs do exactly that. This isn't static information. These are living fingerprints of the ocean's biodiversity. Now, let's look at this box. Here, we see the box plot of sequence lengths. Notice the wide variation. Some sequences are just a few thousand bases, while others stretch beyond 30,000. That spread is exactly why cleaning and filtering are critical. Without it, researchers may drown in noise. Now, let's look at this histogram. This histogram sharpens that picture. A steep peak of short reads, and then a long tail of outliers. This is deep sea reality. Messy data, uneven samples, and hidden gems buried at the edges. The scatter plot reveals another layer, GC content distribution. Clusters from around ribosomal RNAs and known categories. But look at the outliers. Those dots in the fringes, they could be contamination, sequencing errors, or signs of novel life. Now, finally, the bar chart brings clarity. Dominant groups rise sharply, while others remain rare. This tells researchers where biodiversity is concentrated and where more sampling could unlock discoveries. Together, these graphs transform raw sequences into insight, not just numbers, but stories about what lives, what dominates, and what remains unknown in the deep sea. This is how GeneX makes invisible life visible.
Researchers today face problems. Tools like Vega are powerful, but they have complexity. They are overwhelming. They need internet. That's not what every lab has, especially not the remote ones. So we reimagine DNA prediction. From the moment you open the dashboard, everything is clean, focused, and fast. Paste your sequence, hit predict, and that's what you get. You get the predicted outcomes. That's nothing less, nothing more. We built it on JavaScript. Why? Because it's light, stable, and cross-platform, and work completely offline. Perfect for researchers where the internet is limited. It's compared to Vega, which tries to do everything, but our app does only one thing. But DNA prediction, and it does it better. It needs no training, no cluster, but it gives accurate results. And we didn't stop there. We designed the interface like a conversation, simple cards, simple charts, and an AI assistant that explain the prediction. It's not a black box. It's lab partner. Every button, every panel has a reason. No noise, no destruction. Just the tool a scientist needs, right when they need them. It's research simplified. It's not just another research tool. It's the simplest way to get the result, and it's better.
So every solution you are still seeing in this entire demonstration or entire problem statement is all about CNN using keyword, but we did something different. Obviously we did use CNN approach, but we made it effortless, we made it better. What we did is we used LSTM and we went deep into literature and we found out that LSTM is a wonderful solution because it essentially remembers pattern over iteration of training. And what we have to do is generating information in sequential data, and this sequential data is very important. The sequence is very fundamental to the analysis we are about to do, the prediction we are about to make. So we did implemented CNN with LSTM, and it did help us as CNN is the root for all the pattern recognition that we are doing, and LSTM is remembering all the sequences, all the features over time in the model iteration. So looking here, I am trying to import a FASTA file of mitochondrion something, and we are using MAC model. So let me tell about MAC model. So as in problem statement we have given that we need to replace the need of databases. Now, what databases exactly do? They don't predict, they exactly give us the name of the species the sequence is matching. But this is slow. This is very tedious to the system and well to the humans as well, because according to our research, it almost takes 8 to 9 hours to sequence one process and then get the answer for it. So we did something better. We used MAC model, which is our version of that model, and we got a prediction. And if I just hit prediction right now, boom, we get a prediction. And this prediction is, well, probably very fast. And yeah, there would be some issues like overfitting the model. There would be very less stratification of data because the data is not very clean or data is not very stratified or very much segmented to a particular thing. But as we progress, we'll improve the model further. We'll remove this stratification issue. We are actually trying to solve the problem of the novelty detection because there are some outliers, but all of this issue would be resolved in our final MVP product. And the second model we have is Mera Parivar on or who is my family, find my family. So this is a very unique model that we have developed. It finds the distance between two embeddings in the latent space. And why this? Because well, it just uses family members, the nearest family members of the organism, and due to this we can find all the related family members without using the graph neural network or any complex techniques such as, and it is almost same as that, giving us very insightful insights, and that's it. And giving on that, so yeah, that's the thing that I want to highlight is this model is not the best model, but this is one of the cleverest approach we can implement to reduce the load on the system, because this PCs won't be LLMs and LLM supported or having GPUs. So this works on any CPU, any processor, and almost the same speed. So yeah, that's it. Thank you.
So let me show you a fascinating character of this app is having an AI chatbot. So what does this chatbot do? Basically, this research often involves dealing with certain massive datasets and complex visualizations, which can be overwhelming and motile. So this time-consuming process can be avoided by having a chatbot. To make this process easier, we have developed an intelligent integrated chatbot. Like you can see when we say, tell me about a certain species and ask AI, it provides you a proper report, like it's now fetching the AI response. The chatbot acts like a virtual researcher assistant. It can answer queries and also make the guide users through analysis of workflows and explain charts, results, or statistical outputs in simpler and clear language, like you can see. Also, instead of searching through menus, documentation, the researcher can use this chatbot to get a summary and a proper review. The result is faster, insights are better, and also provides a greater understanding of what is happening in this particular AI chat. And also, this provides the user to get a summarized view of the full project. Thank you.
Okay, now let me show you a very useful feature of our project, which is the report generation. So imagine a researcher is working with thousands of genetic sequences. The challenge for him isn't just analyzing them, it's making sense of the results and presenting them in a way that's clear, accurate, and shareable. That's where this feature comes in. So we inputted the header name of the report, and if you want to enhance it with AI, we can enhance. Now coming to the detail part, we will input the name of the species again, and it will generate the description of the species. It is generating right now. So look, who is faster, OpenAI or us? Of course, OpenAI. So it generated. So we can, with just few clicks, researchers can generate comprehensive species reports that are neatly organized and can even be exported with this export to TXT button, with which the file can be exported in the TXT format. So here the best part of the feature is we have designed it for the future ready with AI integration that can add deeper insights and predictive analysis. In short, this feature transforms complex genomic data into meaningful knowledge, which is fast, accurate, and effortless.
Months ago, this was just a problem we wanted to solve. 1 month back, we had working algorithms. And today, we have our prototype ready. We have proven the concept, 80% accuracy. The toughest sequences can be addressed easily. For CMR and RE researchers, this means entire studies in real time. New species are found faster. The ocean has kept its secrets for around 4 billion years. We have built the key to unlock them. Today, we changed how science sees the deep sea. Tomorrow, we rewrite how marine science is.